All right, today's lecture is going to be the second lecture in the nervous system series. And we're going to talk about the spinal cord and spinal nerves. And as a review, and again, you should be drawing this chart and start getting really familiar with it. Oh, that's not what I wanted. Sorry, I thought I had this all ready for you. So we have the nervous system, and it is divided into the central nervous system and the peripheral nervous system. And then the central nervous system has two other branches, which is the motor and sensory pathways. And if you remember, the sensory is afferent. It's going to arrive at the nervous system, central nervous system. And the motor is efferent. And again, it's really important to get used to using these terms and understanding what they mean. And then from the motor pathway, we can take more branches and go via the somatic and the visceral. And remember, what somatic is with motor is that we have control over it. So that is skeletal muscle. And then visceral is what we don't have conscious control over. So that would be cardiac or smooth muscle. And that would also lead us to the autonomic nervous system, right? That's the branch that we don't have conscious awareness of. And we'll touch on the autonomic nervous system just a little bit, but you'll mostly get a lot of that in physio. And then from the afferent, so the sensory, we can break that down into somatic and visceral. Again, practice drawing this out, making sure you know what this means. This is all of our senses like touch, pain, hearing, taste, etc. And then visceral has to do with monitoring, oxygen concentration. We're not consciously aware of that, but our brain is monitoring it. Let's say the pH of our stomach, okay, things like that. All right, so that is the overall view, the chart that we talked about in the very first lecture. You really have to get familiar with this. The peripheral nervous system can also be broken down into motor and sensory. And we'll revisit this diagram as we move into the peripheral nervous system. Right now, we're still working on the central nervous system. So it's a little bit out of order because we're going to start with the spinal cord first before we talk about the brain, because it's a little easier to understand than the brain. So that's why we're kind of working in this order, and that's the order that you'll see in your textbook as well. All right, so let's talk about the anatomy of the spinal cord, the structure. All right, so in an average person, the spinal cord is about 18 inches long. It is actually shorter than the entire vertebral column. So the spinal cord ends between the lumbar vertebra 1 and lumbar vertebra 2. So it's going to end somewhere in between there. And it's going to come to an end. It's going to kind of look like this. I'm going to talk about the structure in a little bit. I'll show you pictures. And then there's going to be spinal nerves that come off of that. All right. And I'll show you that in a minute. So it ends at L1 and L2. And then you don't have a single structure anymore. You have these individual nerves coming off of it. And sorry, I don't know why it does that. The spinal cord is divided into 31 segments. So there are 8 cervical nerves. There are 12 thoracic, and they come in pairs. All right, so there'll be one on each side. There's 5 lumbar, 5 sacral, and 1 coccygeal. So you may be wondering why there are 8 cervical regions, because there are only 7 vertebrae in that region. And that is because there is a spinal cord segment above and below the atlas. So that's what leads up to having 8 segments, okay? All the other ones have the amount of segments that there are vertebrae in that region. Let's take a look at what this looks like here on the next slide. I have a few images. This is the entire spinal cord inside the dorsal spinal cavity. And the bones were removed so that you can see the spinal cord. So you can see the 8 cervical segments right here. And again, they're paired. So you're going to have spinal nerves coming off onto each side. So each side, there's going to be nerves coming off of it. And then there are 12 thoracic, so you can see the thoracic here. There are 5 lumbar here, 5 sacral, and then 1 coccygeal nerve or segment. Now, there are a few features that I want to point out, so we're going to take a closer look at the spinal cord. And it's always important to keep in mind the view in which you're looking at the spinal cord. So which way are we looking at it? So keep that in mind every time you see an image of the spinal cord. So this is the posterior view, right? And this will become important when we start looking at the internal anatomy. All right, so it's always important to have your orientation. All right, so there's a few features that I want to point out. 3 of them are on this side. There's a couple others that I'll point out when I show you a picture of them. So there's a couple enlargements on the spinal cord that are important. So we're going to talk about the conus medullaris. So I was telling you that the spinal cord comes down to an end around L1 or L2, and this is it's a tapered area of the spinal cord, so it's cone shaped, which is how it got its name, conus medullaris, and this is where that lumbar region is, and I have a picture on the next slide to show you about it. So it's at the end of the spinal cord. The other feature is the cauda equina. This is that area where I told you that there's going to be some spinal nerves, individual nerves that come off of the conus medullaris. And these are thread-like filaments that are coming off of the end. And then I'll show you the filum terminale. All right, this is some connective tissue. Let me show you that on the next slide. So this next slide is color-coded, and it's enlarged a little bit so you can see some of these details. So I want to point out a few features for you. So there is a shallow posterior groove called the posterior median sulcus that runs the entire length of the spinal cord. All right, so you'll see it. I'll show it to you on a cross section much later in the talk. And there is a much bigger fissure on the anterior side. So, we're looking at the posterior side right now. You can see this conus medullaris. This is the region where the spinal cord ends in this kind of like a V shape, all right, between L1 and L5. And then the cauda equina means horse's tail, and this is where those nerves are going to come off individually, and it kind of looks like a horse's tail. So let's take a look at what that looks like up close. I go to the next slide. This also highlights the different segments, so you can see the cervical spinal nerves, the thoracic in orange, the lumbar in green here, and you can see the sacral nerves and then the coccygeal. So just to point that out before we move down. All right, so here is a close-up of that conus medullaris and cauda equina region. So what we're looking at is that the spinal cord is actually covered by what's called meninges, and we're going to talk about that in a minute. These are membranes that surround the spinal cord. So you can see these meninges, they are being pulled back, they are resected, and they are cut, and they're pulled away so that we can see the inside of the spinal cord. All right, so you can see the conus medullaris. I'm going to highlight that here, and it forms this nice cone shape right here. And then you can see the cauda equina, which is the horse's tail. Those are all of these filaments that are running down here. These are all nerves that are coming off of the conus medullaris. So one final structure I want to point out on this slide is the filum terminale, sorry. And I'm sorry, this is cut off. It's a transfer when I airdrop this for some reason. So the filum terminale is not actually part of the nervous system. I'm going to highlight what this region is, but I need to change this color to something more dramatic. You can kind of see it here, this piece of material that is running from the tip of the conus medullaris. And what it is a fiber. It is dense, regular connective tissue. So it is dense, regular connective tissue that anchors the spinal cord to the sacrum, because we don't want the spinal cord moving around. That would be really bad. So that is what is going to anchor that spinal cord down and keep it from moving. All right, now I want to take you back and I want to show you a couple other enlargements that are important to note as well. So there's two areas of enlargements that occur on the spinal cord. One of them is called the cervical enlargement, and one of them is the lumbar enlargement, right here. And this is an area of the spinal cord that bulges out. So it happens in the cervical region and it happens in the lumbar region. And this occurs where the nerves are going to the upper limbs and the lower limbs. And they are entering or exiting the spinal cord at these enlargements. And the reason for this is because there are so many nerves that are going to our upper limbs, our arms, and our lower limbs, our legs, that we just need more real estate. We need more space for these connections to occur. So that's why you see these enlargements. All right, and now we're going to take a deeper look at the spinal cord here in this image to get a better understanding of the anatomy. So now you can see the spinal cord, which is surrounded by a vertebra. And you can tell that it's a thoracic vertebra because you can see the costal facets right here. Right on each side. So that is telling you it is a thoracic vertebra. You can also see the spinous process right here. So you know that this is a posterior view. And if you remember when we discussed all of the vertebrae, we talked about how the spinal cord resides in the vertebral canal, all right? And you can also see that there are spinal nerves that are coming off each side of the spinal cord. All right, so they are paired spinal nerves going off to each side. And the spinal cord itself, again, is covered in a series of membranes called meninges. And again, we'll talk about that in the next coming slides. And under the meninges is the spinal cord itself. So that's what you're seeing here. And again, you see the spinal nerves coming off. They're branching off of the spinal cord. And we will also talk about the fact that there is gray matter and there is white matter in the spinal cord. All right, so now let's take a closer look at these meninges. All right, so the spinal meninges, we're going to talk a lot more about them when we talk about the brain, but I want to discuss them a little bit now so you have some understanding about them before we talk in more details. So these meninges are continuous with the brain, okay? They're unbroken, they're continuous. They are the same meninges that you're going to find around the brain. So they are covering all of the tissue in the central nervous system. All right, and there are a series of 3 layers of specialized membranes. All right, so they're going to surround both the spinal cord and the brain. They are going to provide physical stability and shock absorbance. All right, absorption around that nervous tissue. There's also going to be blood vessels within these layers to help deliver oxygen and nutrients to the spinal cord. And then a third thing is that there is going to be cerebrospinal fluid. Remember, we talked about that as well, abbreviated as CSF. This is going to help provide a median to exchange those nutrients and waste back and forth, some gases like oxygen and CO2. All right, so you're going to have cerebral spinal fluid, and I'll show you that more. So there are 3 layers that we're going to talk about, and I'll show you images. And starting with the most superficial layer, you have what's called the dura mater. All right, dura mater, dura means hard, and mater means mother. So this is a very tough, dense, irregular connective tissue. It's very protective. You'll get to see this when we do the dissection on the brain, and you'll see just how tough that is. That is the outside layer. The next layer, which I'll show you, is the arachnoid layer, arachnoid mater. Arachnoid, as you might have heard, is Latin for spider, mother. And this gets its name because there are lots of extensions off of this layer. It kind of makes it look like a spider web. And then there's the pia mater, which means soft mother. And this is the most delicate layer. And it is fused to the spinal cord itself by astrocytes. You remember the foot processes of astrocytes? So that is the 3 layers that I'm going to show you on the next slide. So here you can see the dura mater. All right, again, it's resected here, it's pulled back. And then the arachnoid mater, and I'm going to show you, I'm going to draw a picture here. So if this is the dura mater, I put it as one word, but it's not just because I'm running out of room. And the arachnoid mater is right underneath it, and it has these little struts, and this is what makes it kind of look spidery like, these little struts here. These are called trabeculae. Do you remember that word when we studied bone? So those are the trabeculae. And this is arachnoid motor. So these two membranes are very closely associated with one another. And that layer will connect with the pia mater. So you can see they're somewhat close together. They're right on top of each other. And there is a space that is created between the arachnoid mater and the pia mater. All right, this hollow space contains the cerebral spinal fluid. So that cerebral spinal fluid is going to run in between that space there. And there's also going to be blood vessels. And then the pia mater is fused to the spinal cord and it follows the contours of it. So here is the pia mater. Here you can see that it is fused to the spinal cord. All right, so these spaces are going to become really important. All right, so let's take a moment to talk about these spaces because they have some clinical significance to them, which makes them important. All right, so again, and I really need you to understand these spaces and to know which layers form these spaces. So let's talk about the subarachnoid space. So again, this is between the arachnoid mater and the pia mater, and it is filled with cerebrospinal fluid, and there are also blood vessels there. And again, this is going to act as a shock absorber, and this provides a medium for gases to be delivered, so like oxygen, and then CO2 can be exchanged. You're going to get some nutrient exchange there as well, and then some chemical messengers and waste can be removed. All right, there is one other space that I want to talk about, which is called the epidural space. And let me show you what this looks like so you can see. Now, this is a true cross-section of a spinal cord. Okay, so you can see the spinous process, right? We always want to orient ourselves. Here's the spinous process, so you know that this is the posterior side. You can see the anterior side near the vertebral body. All right, so the subarachnoid space, so we were just talking the dura mater is most superficial, okay? Then you have the arachnoid mater, and then you have the pia mater. So we already talked about the subarachnoid space. Subarachnoid, that's one word. Space, right? All right, and then and in that subarachnoid space is where cerebral spinal fluid is going to be. So there is also a space between the periosteum of the bone. So here, let me get something that will show up. Here is that periosteum of the bone right here, right? That's the periosteum of the bone. And then there is a space between the periosteum of the bone and the dura mater. So the dura mater is right here. I'm trying to draw this as exact as I can. It's really hard on the iPad. And there is a space right there called the epidural space. Epi meaning above the dura, epidural space. And this is largely filled with adipose tissue, and it serves as a cushion. That's what you're seeing in these pictures. So why are these two spaces important? So clinically, you've probably heard of an epidural, right? So women who are delivering babies often get an epidural, and so this space becomes important because we can deliver a drug into this epidural space. Now the subarachnoid space, I'm going to show you a picture of this. All right, so that's the epidural space, and I'm going to show you why that's clinically important as well. And then the subarachnoid space, that is where the cerebral spinal fluid is flowing. So there may be an instance where you might need to obtain a sample of cerebral spinal fluid, okay? And maybe, there's an infection or something that you need to sample that fluid. So we need to be able to insert a needle into that space. So let me show you what that looks like. All right, so again, there's a lumbar puncture and then the epidural. So in the instance of the lumbar puncture, and that is because we need a sample of cerebrospinal fluid, we need to insert a needle that's going to break through the dura mater and go into, so it needs to enter the subarachnoid space, okay? I was just gonna write that out, but I see it right there. And that needle is very close, like it's very sharp, and it could be very close to the spinal cord. So if it punctures the spinal cord, it's going to sever neurons. And therefore, there's a technique to do this lumbar puncture between L3 and L4, sometimes between L4 and L5. And do you remember why this is? So you remember how the spinal cord ends, right? And that usually happens between L1 and L2. And then after that, you have those individual nerves coming off of there, right? So typically, if you're going to do the puncture, you want to do it lower than the spinal cord. Because if you stick a needle in here, okay, there will be less likely to injure the spinal cord because you're not in a region where the cord is anymore. These are individual nerves. And what can happen is if you do like a cross section and you are looking at these nerves, if you stuck a needle in here, the needle may just push aside some of those nerves so that you can then obtain the cerebral spinal fluid without injuring the spinal cord. So you are less likely to harm a patient if you do it where the cauda equina is rather than where the spinal cord is, okay? Now, the epidural is done to deliver anesthesia. So if you deliver it to the spinal cord, that would anesthetize the entire central nervous system. And that would be really, really bad because cerebral spinal fluid goes everywhere. So that wouldn't be good. So what we need to do is we need to deliver it into a space where there is not a lot of, where there isn't any cerebral spinal fluid, but rather deliver the drug into an area that cannot gain entrance to the cerebral spinal fluid. It's going to leak onto some of the nerves, and actually the sensory nerves are more sensitive to the epidural drug than the motor nerves are. So what ends up happening is you remove the sensation of pain, but you're still able to push with your muscles. So you still have motor ability. So I think these pictures show it nicely how the needle is being delivered into the epidural space. So if we go back here, the epidural is going to be delivered in that space rather than the space that contains the cerebral spinal fluid. Okay, now let's take a look. Let's talk more about the nervous tissue itself, okay? So we are going to look at how the nervous tissue is divided. And you're going to see on the next slide when I show you a cross-section of the spinal cord up close that the tissue is divided into two regions. So you're going to see white matter and gray matter. Okay, the most superficial nervous tissue is the white matter, and the gray matter is deep to that. The white matter consists mostly of myelinated and unmyelinated axons, if you remember from the first lecture. And it is going to contain both ascending and descending tracts, nerve tracts. So the ascending tracts are going to the brain. And we know information going to the brain is most likely sensory. And then we have descending tracts that are going from the brain. And these are most likely going to be motor tracts. So ascending is going to be sensory tracts, and descending is going to be motor tracts. And these are all of the axons going in these different directions. Gray matter, on the other hand, is dominated by the cell bodies, or the soma, of neurons. It also makes, and there's also neuroglia in that area. So remember that the neuron cell bodies are the cell itself, and then the axons coming off of that, that's what's making up the white matter. So the cell bodies are all clustered together, and they cluster together based on similar functions. And these clusters of function, like the cell bodies that have similar functions, they're clustered together, and unfortunately, I wish I could have changed this term, they're called nuclei. It's not a nucleus like a cell. It's unfortunate that they use this word, but it is a group of neuron cell bodies. So also called the nucleus. So nucleus, and again, it's unfortunate that this term is used, but this is a group of neuron cell bodies. They have similar functions, and they are in the central nervous system. I will go on to show you that the term ganglia is a group of neuron cell bodies in the peripheral nervous system. So let's get used to using these two words. So again, the gray matter is dominated by cell bodies of neurons and neuroglia. They are organized into functional nuclei. So they are organized by their function, and they are grouped together. They surround a small central canal. Remember that the cerebrospinal fluid runs through the central canal, and the gray matter often looks like a butterfly or takes on an H shape. So let's take a look at that. All right, so this is a cross section of the spinal cord. So you can see this posterior median sulcus that I talked about in the very first slide or two. There is a very deep anterior median fissure. The white matter is superficial. So let's see, all of this area right here, this is all white matter. And the gray matter is deep. It takes on a butterfly shape or an H shape. And it's worth noting that this orientation is in complete contrast to the way that the brain is structured. So in the brain, the gray matter is superficial and the white matter is deep. You can split the spinal cord kind of down the center, if you will, and each side of the spinal cord, the left and the right side, are able to communicate with each other. And let me introduce you to two new terms. So there are tracks. Let me change this color. There are tracks that run across the center here. This, for gray matter, it is called the gray commissure. And there are also tracks on the white matter. That would be the white commissure. And what these do is that they allow for communication between the sides. So signals can cross over from the right side to the left side or from the left side to the right side. And if you remember from our very first lecture, we talked about contralateral. Do you remember contralateral? We also use the word ipsilateral. So we're going to now bring that term and use it. So this means from one side to the other. So there can be communication from the right side to the left side. Another term for this is decussation. So the ability for the left side to communicate with the right side. We're next going to talk about how the white matter and gray matter is organized. All right, so we're now going to talk about how the gray matter is organized, and then we'll move on to how the white matter is organized. And we're going to talk about how it's organized in a functional way. Okay, so I showed it to you before we kind of looked at the anatomy, but now we're going to see exactly what is going on. So the functional organization of gray matter is broken up into two categories. We're going to have sensory nuclei and motor nuclei. Now, the cell bodies of the neurons, right, are going to be grouped together functionally in the gray matter of the central nervous system. And again, we use the term nuclei when we're talking about groups of cell bodies in the central nervous system. When we start talking about the functional groups in the peripheral nervous system, we're going to use the term ganglion, all right? And again, these are grouped into sensory nuclei. These are going to receive and relay all of the sensory information. And then there's going to be motor nuclei, which are going to be grouped together also to issue motor commands. All right, on this next slide, I have a lot of terms here, but what I want to do is take you to a picture of it first, and then we'll look at the terms, and it should all make a lot more sense. So this is a cross-section, again, of the spinal cord, okay? And what we want to do is immediately orient ourselves to posterior and anterior. So you can see here is the anterior median fissure, and we can make out the posterior median sulcus. So again, it's important to always orient yourself so you know which direction is which. All right, so now that we have orientation, we're going to take a look at the gray matter. So that's the deep neural tissue that looks like a butterfly or shaped like an H. That is the material that we're going to talk about, and we're going to organize this. So first, let's organize it into sensory and motor, and then we'll break it down even more. So everything on the posterior side is sensory information. So everything here is sensory, and everything on the anterior side is motor. Okay, you can see that here. And we can also divide, so that's this dividing line here. We can also divide it into left and right. And remember, we talked about if when the left and the right side communicate with each other, they're going to communicate through the gray commissure if it's gray matter, and the white commissure if it's white matter. Then we can further break down the sensory information into somatic versus visceral. And there are distinct locations. The nuclei are grouped together based on the type of information that is going to be received. So all somatic sensory information, all the nuclei are going to be clustered in this region. And all of the visceral sensory nuclei are going to be clustered in that purple region. Now, I don't want you to memorize these different regions. What these images are showing you is how complex and how organized the spinal cord is. And it's such a sophisticated mechanism by which all of these nuclei are arranged. Now remember what somatic sensory is. That's the ability for us to interpret touch or pain. And then the visceral sensory has to do with pH or oxygen levels. So both the somatic and the visceral sensory nuclei are located in what is called the posterior gray horn. And again, there will be connections that run between them, back and forth from the left to the right side through the gray commissure. Now, the anterior half of the spinal cord is all motor nuclei. So that's here. So you can see this cluster of light blue. That's where all of the motor nuclei, or the nuclei that will receive or send out visceral motor information. And then you can see in pink the visceral somatic motor information. So both the motor nuclei are divided as well into visceral and somatic, so that they're organized in a very particular way. So remember that somatic motor would be, let's say, the bicep brachii muscle, right? So there would be axons that will travel toward the bicep muscle to have that muscle contract. Whereas the motor visceral would be going to like the autonomic nervous system, so our internal organs, things that we don't have control over. Now, the visceral motor nuclei are located in the lateral gray horn, and the somatic motor nuclei are located in the anterior gray horn. So there's names for all of the locations of these nuclei. So let's take a look back at this slide, and this slide summarizes where those nuclei are located, and it's showing you the organization of gray matter. So the posterior gray horn contains somatic sensory and visceral nuclei. The lateral gray horn contains visceral motor nuclei. The anterior gray horn contains somatic motor nuclei. And then the gray commissure allows communication back and forth from one side to the other. And that is what I outlined here. And it's showing you the posterior gray horn, the lateral gray horn, and the anterior gray horn. Now let's look and see how the white matter is divided. The white matter is divided into columns on each side of the spinal cord. So there's what's called the posterior white column, there's a lateral white column, and an anterior white column. Each of them contain tracts, and these tracts are simply bundles of axons. So all axons in the tract relay similar types of information. So they're either going to relay sensory information or motor information. They are never mixed. They can communicate through the white commissure. And remember, the ascending tracts are always going toward the brain, and that is sensory information. And the descending tracts are always motor information, and they are descending. So let's look at a picture of that. So here is the white matter, of course, and all of the information is organized into these what's called white matter tracts. So the sensory information is going to the brain from each particular region of the body. And again, this isn't meant to necessarily for you to memorize, but appreciate how detailed and precise this needs to be. So you can see how here in this pink color, this is information that is coming from, because again, sensory information is here and motor information is here. So sensory information from the leg, the hip, the trunk, the arm, it all has a very specific place on the spinal cord where that information will get relayed back to the brain. All of these regions are then categorized. So you have the posterior white columns, you have lateral white columns, and you have anterior white columns. And all of those have specific regions in which they are communicating with the rest of the body. Now let's take a look at spinal nerves, because you can see that they're starting to come off of the spinal column. So we're going to switch and start integrating the information on the peripheral nervous system and how these spinal nerves come to be. So again, spinal nerves are part of the peripheral nervous system. Every segment along the spinal cord has a pair of peripheral nerves. So there are 31 pairs of spinal nerves that come off of the 31 spinal cord segments. And they are identified by the association with the adjacent vertebrae. So where they are in relative to the vertebrae. For instance, there are segments off of the cervical region. There are segments off of the thoracic region, the lumbar region, and the sacral and coccygeal region. So there's going to be a pair. Every segment is going to have a pair of spinal nerves that are associated with that region. All right, this is a picture showing you all of the regions, the spinal cord regions, and there are pairs that come off on each side. They're going to come off on each side off of each of these segments. The spinal nerves contain axons that contain both sensory and motor neurons. And there are roots that are coming off of the spinal cord that are going to lead to these spinal nerves. The roots coming off of the axon, off of the spinal cord there's two of them. There's a ventral root and a dorsal root, and they contain very specific information. The ventral root is always going to contain motor neurons, and the dorsal root is always going to contain sensory neurons. And I'm going to come back to this dorsal root ganglion when I show you a picture of all of this. All right, so again, let's orient ourselves. Here is the anterior side. Here is the posterior side. And off of the spinal cord are two roots. You have the dorsal root and the ventral root. So neurons that reside in the spinal cord, their axons are going to travel through these roots. The dorsal root is always going to contain sensory information, and the ventral root is always going to contain motor information. So remember that all sensory information is going to arrive at the spinal cord to be processed. It can then be sent up to the brain for interpretation. And then motor information, the efferent pathway, is going to leave. So axons are going to travel through the ventral root and make their way toward this spinal nerve where both of them are going to converge. There's a feature in the dorsal root called the dorsal root ganglion. So what is this? This is a group of neuron cell bodies in the peripheral nervous system. So now it becomes a ganglion. And if you remember, all peripheral sensory neurons are pseudo-unipolar. If you remember what pseudo-unipolar neurons look like, the cell body is off to the side. And this is where all those cell bodies are going to reside. They are collected here in this dorsal root ganglion. So the cell body is in this dorsal root ganglion, and its axon is going to stretch all the way through into the spinal cord. Whereas the motor neurons are going to reside here, I can't really draw it, and their axons are going to come out the ventral root and head toward the rest of the body. This is how you connect the spinal nerves to and from the spinal cord. Before we get into the heart of the peripheral nervous system lecture, I do want to go over the histology that you will see of the spinal cord in the lab. So now that we've talked about a lot of these features, you can make sense of it when you look at a histology slide of the spinal cord. So let us orient ourselves as we always do. So here is the anterior median fissure. I'm going to highlight this. And then the posterior median sulcus. If ever you're having a hard time orienting yourself, you can also find the dorsal root ganglion, right? It's this bulge right here. And remember that dorsal means posterior, and ventral is anterior. So you have the ventral root and the dorsal root. That's a structure to look for if you're trying to figure out orientation. If we look at the gray matter, we can organize the gray matter. If you remember, we have the posterior gray horn, we have the lateral gray horn, and we have the anterior gray horn. One feature that I forgot to mention is that in the center, you have the central canal. And remember, that is where cerebrospinal fluid is going to flow. And then we can also, and although it's not listed on here, we can also organize the white matter. The white matter was organized by columns. So you could point here, and this would be the posterior white column. This would be the lateral white column, and this would be the anterior white column. You can also notice the gray commissure that's going to run here. This would be the anterior gray commissure. This would be the posterior gray commissure. There's also going to be white matter white commissure here and here, anterior and posterior, and you can begin to make out the meninges as well that surround the spinal cord. We talked about the dura mater, the arachnoid mater, and of course the pia mater is closest to the spinal cord. So that is what you should look for and be able to identify when you're in lab, the histology of the spinal cord. I think this picture best summarizes what I just went over. And it's showing you with a little bit more detail exactly how this is working. So I wrote on here to just orient us anterior versus posterior first. And remember that all sensory information is on the posterior side and motor information is on the anterior side. Because I want to stress how important this, the ganglion is. The dorsal root ganglion right here. So we have sensory information. So remember that all of these neurons are pseudo-unipolar. So this is a great example. You can actually see the neuron here. You can see how the cell bodies are going to cluster and form that dorsal root ganglion. So all of the sensory information, so this is out in the periphery. And this is where the receptor endings are. So if we get, let's say our finger touches something sharp, that information will travel down this pathway. So you can follow the neuron here. All right, and there's the cell body, and it's going to enter through the dorsal root. That information is going to, that neuron's going to synapse with another neuron, remember the interneuron. That information can then go up to the brain for analysis and then back down. And then we can get a motor response with another neuron. So here is the other neuron. The cell body is residing within the gray matter. And then its axon travels through the ventral root. And then you can have an attachment of that axon to a specific muscle, skeletal muscle, to then move your finger out of the way. So I feel like this is a good summary of how and why everything is structured the way it is and why you see these dorsal root ganglion. It's a great way when you're looking at the histology to recognize the dorsal root ganglion, because that can orient you immediately to posterior and anterior.